Thank you, everybody, for being here on this fourth of our conversations between Instituto de Salud Carlos III and IARC, Instituto International Agency for Research in Cancer. So it's my pleasure to have these eminent people here talking. And I'm just going to pass the microphone to the chairman today, who is Maria Jose Barrero. She is a scientist she just incorporated into our institute, particularly in the Rare Disease Institute. And she's also an expert in epigenetics, so she's better than me to present this fourth seminar on epigenomics and mechanisms of cancer. So, Maria Jose, the microphone is yours.
Thank you very much, Pilar. Today we have two great speakers, experts on the field of epigenetics and cancer. First speaker is Dr. Zenko Ekser. Thank you. He's the head of the branch of epigenomics and mechanisms at the International Agency for Research on Cancer in France, where he has been working since 1999, holding different positions. He has an international reputation in the application of epigenetic and epigenomic analysis and profiling studies of tumorogenesis and on the discovery of biomarkers of environmental exposures and cancer risk. He has a remarkable publication record with more than 200 original peer-reviewed papers, mainly in the field of epigenetics and cancer. So all these publications have been cited more than 16,000 times. He has been an invited speaker at over 150 international conferences and research institutes. And he is also very active at initiating and developing international collaborations that have been funded by major international and national funding agencies. Not only that, but just yesterday, I found out that he is also an artist. And I would recommend that you follow his Instagram account at Scientist Painter, where you can find a very interesting combination of art and science. His lecture today is going to be focused on epigenetics and environmental origins of cancer. And we are looking forward to learn about the state of the science of epigenetics, how environmental stressors impact epigenetics status and are linked to cancer risk. Also, we will learn about technological advances in epigenetics and also on the identification of epigenetic targets that could be used for personalized medicine, but also can be applied to the field of molecular epidemiology. So, Zenko, thank you very much for joining us today. We look forward to your talk.
Thank you very much, Maria, for this nice introduction. I would like to start by thanking the organizers, our friends from Carlos, from Instituto de Salud Carlos III, Pilar Sanchez Gomez, who we know from the Scientific Council, and of course Manuel Esteller, who is also an important figure in this field, and a colleague, a very active colleague and collaborator. And I would like to just also mention that in my presentation there will be two other speakers who are in my branch. Branch is called Epigenomics and Mechanisms branch at IARC. Doctors Julie Zavadil, who is deputy head, and Rita Query, who is scientist. So, without any further ado, just to give you a short introduction on IARC, but just to basically a couple of slides that we are positioned. Our branch is actually one of the 8 scientific branches, as you can see here, these are the structure of our branch. This nicely structured, well representation of these, senior, mid-career, and early career scientists and supporting staff. Overarching goal of our research is to advance understanding of cancer causes through investigation of both epigenetics and genetic patterns that are regulated by environmental exposures. And I'm using environment here in its broadest definition, and understanding the underlying mechanism. And this is actually to enhance evidence base for cancer prevention, but also early detection and therapy. We all know that most cancers are driven by environmental exposures, different risk factors for endogenous or external sources or endogenous processes that regulate genome and epigenome. And these changes actually are fueling this what's called hallmarks of cancer. And when they're regulated, can promote the tumor cellular transformation and cancer development. So therefore, looking at the epigenetic changes, actually we can look at the sensors of exposures, but also mediators of the outcome. And important, the aim is to kind of identify causal pathways that link these exposures to cancer and provide biological plausibility to the associations that are often detected in epidemiological settings. So these are the major aims, is to identify epigenetic patterns of exposures, and also to identify potential markers of exposures or cancer risk. Also, we are following often this profiling study with identifying causal pathways that link this environmental lifestyle exposures to tumorigenesis. And also we develop epigenomic or genomic methodologies and profiling strategies that are applicable to the large population-based cohorts and biobanks associated with this, studies that are coordinated by IARC and our international collaborators. This is a kind of our strategy, using the population-based cohorts or case control study, more recently intervention studies, often combined with either cellular systems or even mouse models. We take advantage of this spectacular progress in technology. Often we link our data with the data repositories in public domain. We validate our finding and follow when necessary with the focused studies in vitro to kind of provide a functional meaning of the changes that we identify either in cancer or surrogate tissues. This is one example of the EPIC cohort. That's the large European prospective cohort on diet and cancer. And the biobank was quite impressive, biobank that IARC actually house. We also promote setting up international consortia. This is one example of international childhood cohort consortium, a cancer cohort consortium that actually regroup many national mother-child cohorts. And also we develop methodologies that are applicable with different types of samples that are collected as a part of this consortia and national cohorts. We develop methodologies, wet lab pipelines, as I said, and for example, metallome profiling, for chromatin analysis, RNA profiling, but also mutational profiling. We have a robust pipelines in-house, but also we outsource when needed for high throughput, large-scale sequencing. And of course, this is combined by bioinformatics tools. This is absolutely essential to be able to analyze, interpret large data sets. And we have also bioinformatics pipelines and the people that actually has the competencies and the tools that can help us to analyze the data, large data sets. This is just an example of this international collaboration where we profiled the epigenome in esophageal squamous cell carcinoma. That is one of the major cancer with unknown etiology in different belts, for example, East African belt. And as you can see here on the on these circles plots across chromosomes, you can see how methylome is heavily deregulated, and in both directions, hyper or hypo. So the challenge is really to identify what are those functionally important drivers, and what are the early changes, but also potential markers that can be used, for example, in minimally invasive specimens, like cytosplon shown here. And we are trying to work with the collaborators to see whether some of those early changes that we identify in esophageal squamous cell carcinoma can be detected in low resource settings. So another example, we try to look at different layers, and this is, for example, cross-omics analysis of cutaneous and acral melanoma. This is also taking advantage of our expertise, but also international collaboration, more specifically in Brazil, and where we can actually identify not only specific changes and signature associated with UV or other risk factors, but also identify the prognostic markers or the outcome. One of the major effort is really to try to understand causes of childhood cancer, and as you know, the epigenome is undergoing the rapid reprogramming during in utero life, and therefore a hypothesis is that different exposures during in utero life can be fixed and propagated through the epigenetic mechanism, and can contribute to cancer risk in childhood, but also in later life. So for this, we take advantage of large and unique prospective cohorts. And so some of them are, in the interest of time, I will not list them. Some of them are really impressive, with around 100,000 mother child pairs. And also combined with our technology, methodology that allow profiling at birth and neonatal samples, both cord blood, but also blood spots. Just to give you one example, extreme example, how the exposure during utero life can impact the epigenome, and these changes can be detected in neonatal samples. And this is a maternal smoking, and this is well known and reproduced by many groups. As you can see here, different top hits that are associated with maternal smoking during pregnancy. This, of course, is debated. Is this kind of causally influence the risk, but regardless of this debate, the fact that we see such a strong, robust markers suggests that looking at the epigenome, actually we can identify potential causes, known and suspected causes of cancer. We look, of course, at the methylome as a potential markers of risk that can predict cancer, not only that can actually tell us about history of the exposures. And so we hear some exciting data that we can see at birth that may predispose to childhood leukemia risk. Another area is more on the adult cancer, that we also try to take advantage of prospective cohorts, but also intervention studies, where we want to see whether the epigenetic changes and perhaps other layers like metabolome or microbiome, whether they can actually impact be in the causal pathways, mediating pathways to predisposing conditions like obesity or metabolic disorders, and also to see whether we could intervene and take advantage of this intrinsic epigenetic reversibility. So, for example, by weight loss, metformin treatment, and other interventions, whether we can reduce, whether it can reverse epigenetic changes and reduce cancer risk. And we are focusing here mostly on the breast cancer and colorectal cancer. And obviously, the ultimate aim is to identify markers that can actually be integrated in this kind of more personalized early detection, prevention of different cancer types. So now, apart from this more biomarker profiling and identifying biomarkers and of risk and exposures, we are trying to gain insights into the mechanism. What is the functional importance? And so this is one project, what we call epi drivers, that are kind of pan-cancer approach, where we have a pan-cancer genomic and experimental strategy to identify and characterize the epidrivers in cancer, and also their link to the environmental exposures. And I think I will just now switch to my colleague, Rita Query, who will present briefly this project. And later we'll have Jiri Zavadil, who will talk about his focus.
Thank you, Zdenko. Thank you all. So today I will, as Zdenko mentioned, talk about one project that we developed in our branch, and we call it Epidriver Project, with the aim of revealing epigenetic drivers of cancer. We call them epidrivers, and they're linked to environmental exposure. So because we noticed that a high frequency of the regulation in epigenetic regulator genes in tumors, and because, of course, we all know the key role that epigenetic mechanisms play as a link between the genome and the environment, we are convinced and we hypothesize that epigenetic regulator gene may act as drivers of cancer onset and progression and phenotypes, and the deregulation of those epigenetic regulators result in a chromatin rearrangement, enhancing carcinogenic potential of environmental exposures. So, as I mentioned, the objective of this project is identify and characterize the main epidrivers of several cancers and test their role in potentiating the genotoxic and non-genotoxic agents in tumorigenesis. So basically, we developed a conceptual framework allowing the identification of candidate and epidriver across different cancers or specific to certain cancers. And this is based on an in silico approach and in vitro approach that I will talk about a bit later. So our in silico approach is based on data curation from clinical samples. We use, for example, TCGA database of 10,000 samples to curate expression, epigenetic and genetic alterations in 426 epigenetic regulator genes from different classes that regulate the whole epigenome. So the idea is to perform an intra and pan-cancer investigation of those alterations, and then intra and cross-omic analysis that will allow us to identify the driver potential of those genes. So I'm just giving some examples of the analysis that we performed. So we looking at the genetic alteration, we looked at mutation, amplification, deletion, and what we noticed is a high percentage of alteration in those epigenetic regulator genes across 33 cancer types, as you see here. And then some of them, for example, for ovarian cancer, we have over 50% of samples representing at least one alteration of genetic alteration in one epigenetic regulator gene. Then we went to identify the highly altered, genetically altered epigenetic regulators per cancer type. This is just an example of one type of genetic mutation, which is copy number variation, but we did this for all type of alterations. After looking at genetic alteration, we went to look at expression as well of those genes. And then we saw as well, again, a high number of those genes altered at the expression level in different cancer types. And then we identify per cancer type the highly deregulated genes. Interestingly, when we look deeper at those genes, the majority of those genes are actually upregulated. Then we used all those investigation and curation, and we developed algorithms allowing to give the driver potential of those genes. So here I'm just showing one example of this, of the results of those algorithms that we developed, which is the top 100 epigenetic regulators that have the highest potential driver pan-cancer, meaning they are altered in more than one cancer. But we did this as well per cancer type. And I will show, for example, for breast cancer later on. Then after having all this in silico data, we know that this is actually a snapshot of what's happening in the tumor at a certain time. And our aim was to go deeper and to understand, to identify and validate in vitro specific epigenetic drivers of cancer onset, development, or phenotype. So we developed this in vitro approach based on CRISPR-Cas9 screenings and CRISPR activation that will allow us to identify those genes. So basically, we have normal and cancer cells on which we apply this CRISPR-Cas9 screening library that is targeting 426 epigenetic regulator, and then we do a phenotypic screening. I will give some examples. For example, we identified epidrivers of the epithelial to mesenchymal transition in lung cancer. Several genes we identified, and then as well, interestingly, several of those genes were altered in our in silico approach. And then we went to validate as well many of those genes. I'm giving one example of one gene here, EP400, that plays a role in a complex regulating histone acetylation. And then we see that indeed, when we knock out these genes in epithelial lung cancer cells, we have a loss of epithelial marker, an increase in mesenchymal markers, and then we have an increase as well in the wound healing capacity of those cells. So confirming this as well, migration, increase of migration, which is usually linked to EMT. Then, after validating our in vitro approach using those lung cancer cell lines, we went to study a bit more in depth breast cancer. And then we wanted to identify epidriver of breast cancer cell plasticity and study their link to endocrine disruptors. So we went back to our framework, and we did in silico analysis, and then we studied the regulation of epigenetic regulator genes in all breast cancers, and then in subtypes of breast cancer. Here I'm showing the algorithm of the that we developed that allow us to identify the top epidrivers with epigenetic regulator genes with a high driver capacity in breast cancer. And then we used our CRISPR-Cas9 screening in vitro to identify epidriver of breast cancer cell plasticity, focusing on breast cancer stem cell-like phenotypes. And then what we aim now is to study how those epidriver will enhance the impact of endocrine disruptor in breast cancer development. We're doing this in 2D culture, but as well we developed organoids and tumoroid, and we would like to study this as well in 3D models in breast cancer. This framework, we used it for breast cancer, but as well we are using it to understand the epidrivers of EBV-induced Burkitt's lymphoma and their link to mycotoxin. This is in the context of a project that we have that aims to study the interplay between mycotoxins, infectious agents, and epigenetics in the development of endemic Burkitt's lymphoma in sub-Saharan Africa, where we are studying a cohort of the epigenetic in a cohort of blood from children in Africa. And then coupled to this, we have as well in vitro approaches where we study the epigenetic regulator gene deregulation throughout the process of EBV-induced B-cell immortalization and chromatin as well landscape changes. So to even go deeper into this analysis and have a bit more mechanistic understanding, we used our in vitro CRISPR-Cas9 screening. Here, we would like to use it to understand or to identify the epidrivers of this immortalization of primary B cells by EBV, but as well with mycotoxin exposure. So we hope, and we are trying to identify epidrivers of cancer development and aggressive phenotypes using this approach, and then establishing new strategies for carcinosin evaluation, and then as well new strategies for epigenetic-based early detection, prognosis, and personalized prevention, and as well treatment for different cancer type. Hoping soon as well we will use this approach for glioblastoma. So many people of our branch are involved in this project, and then with many collaborators, we hope to achieve our outcomes. I will stop sharing and hand it to Jerry.
Okay, so first of all, thanks for the opportunity to be part of these exciting conversations. I will present our work. The title summarily calls the presentation Toxicoepigenomics of Cancer Risk Factors. Essentially, we developed an integrative screening design, which combines different systems for molecular analyses, and those are in vitro-based, in vivo-based animals. We also analyze human samples, and then we analyze pan-cancer genome data. And we are looking for the effects of carcinogens, of either established or candidate carcinogens, carcinogenic factors. And the approaches look at cytotoxicity, genotoxicity, mutations, DNA-ARC formation, mutational signatures, but also non-genotoxic effects such as epigenome alteration, chromatin structure, and so on. And the idea is to generate mechanistic data for each tested compound which would allow it to basically to be classified in terms of the key characteristics of carcinogens. Let me try something here. The pointer. And then these mechanistic data support the carcinogen evaluation classification by IARC, but ultimately leads to cancer prevention measures just by understanding the causes. And we, in principle, are trying to expand this current map of cancer sites linked to specific cancer risk factors. And our roadmap for testing originates from the recommendations of the advisory group recommendations on priorities for IARC evaluations of carcinogens. And here you can see some of the prioritized compounds, with the yellow ones highlighted being those that we work on. And this approach can be then used in molecular cancer epidemiology studies, integrated toxicogenomic studies, where we focus primarily on mutational signatures as the biomarker of exposure. A couple of vignettes. One study that we published some years ago focused on the mutational signature of aflatoxin, which hadn't been well defined at that time. And we again applied this multi-system screening approach. We sequenced genomes, exomes from exposed cells, exposed animals, and also from the tumors of exposed people from a region of high exposure in Shidong, China. And we were able to identify mutational signatures of aflatoxin, which were kind of similar across the systems, but they also differed with some details between the experimental and the human parts. And this then allowed us to perform a vast in silico screen for the presence of that signature in liver cancers across the world, thereby identifying regions of high exposure prevalence and also low exposure prevalence. And we think that this is a good basis for any future studies where the exposure to aflatoxin will actually persist due to climate change conditions and so on. Another example where we applied this integrated multi-system approach is acrylamide from the diet, which is a compound created in heated starchy foods. And acrylamide generates glyceramide, which is a reactive metabolite. And again, we had the multi-system approach. We looked at cells, animals, and humans. We defined the signature of glyceramide as the readout of the acrylamide exposure, identified its distribution in different populations of the world, looking at the peacock cohorts from the ICGC. And the highest signal came from kidney cancer, which was quite interesting. So that signature is present in kidney cancers quite highly across the world. And that fits with the previous epidemiological studies, which looked at the relative risk of dietary acrylamide exposure and renal cancer risk. And you can see there's a 50 to 60% increase here. And then we contacted colleagues at the Maastricht University who have renal cancers from the Netherlands cohort study on diet and cancer, which also looked at acrylamide exposure. And we are currently sequencing genomes from low exposure and high exposure patients. And we already have some first signatures coming out, and we are also mapping specifically our glyceramide signature on these genomes. So this is a currently work in progress. And then obviously this approach can be applied to many other exposures. So we also used it for studies of aristolochic acid, recently reviewed in Nature Reviews Cancer, asbestos and ovarian cancer, tobacco-specific nitrosamines in smoking-associated cancers, various mycotoxins, and recently we started working on acetaldehyde and oral cancer. The second part of my talk will provide some examples of how we can actually model cancer mechanisms in vitro, looking at either driver mutation events, but also important epigenomic regulation mechanisms. So one of the systems that we use in vitro is a very old one, but it's actually quite an interesting. So these cells, primary fibroblasts from the mouse, mouse embryos, they actually undergo spontaneous immortalization, which goes through senescence stage, and then bypass of that senescence and expansion of a clone. And we realized that we can use the feature of this culture to study different processes that are related to cancer. And for example, we exposed these cells at the baseline level to many different mutagens. We were able to generate mutational signatures, which actually mimic the signatures seen in human cancers. And then we also saw that there are recurrent mutations which actually correspond to driver mutations in humans. An example here is the oncogenic RAS codon 61. And we were then able to even go a little bit deeper and look at combinations of mutations and to do some validation testing for identification of these events as true driver events of the immortalization process. So the driver events can be modeled in vitro, and we published that some time ago. And then the second thing that we looked at more recently, in those cells, we actually engineered the SMARCA5 chromatin remodeler gene, and in such way that we could delete either one or both alleles. And you can see the hemizygous versus the homozygous deletion here. And when we compared the hemizygous with the heterozygous, we could see some effects on cell proliferation. So this would be the conditional knockout of both alleles or one allele. You can see the drop in proliferation capacity. But more importantly, the presence of SMARCA5 also ensures fitness of the cells. And if the cells are challenged by mutagenic genotoxic insult by one of these compounds, you can see that in the absence of SMARCA5, you see less viability as shown by these two lower lines in either case. And this was recently published. And more importantly, the conditional knockout actually affects the overall process of the immortalization. So in the hemizygotes, there's only a partial immortalization as measured on 100 cultures, immortalization assays. But where we remove both alleles, only 8% basically immortalized. So SMARCA5 is important for proliferation, protection against genotoxic insults, but also the spontaneous immortalization. And as such, it represents an interesting cancer gene. And then we also looked a bit deeper at the mechanisms. So following the gradual decrease of the levels of SMARCA by the hemi versus homozygous deletion, we actually looked at the corresponding ATA-XC, RNA-XC data and identifying thousands of differentially accessible sites. And this was the same case for the opposite direction. So we actually have also the programs explaining the phenotypic processes. So here I would just summarize that for the first part, the multi-system toxicogenomics studies help us understand the exposure impact in humans and also to identify biomarkers of exposure and in some cases of cancer formation. And then some of the in vitro system using the multi-system screen such as the primary cell systems. They allow us to investigate key molecular underpinnings of the cancer process. And with that, I would like to thank all the colleagues in the branch and the team that I work with, all external collaborators and our funders over the years that supported this work. Thank you very much for your attention.
Okay, thank you very much. Those were great talks. I also appreciate that you have adjusted the time. So we are on a schedule, and we will move the questions to the end of the next talk. Next speaker is a very well-known leader on the cancer epigenetic field, Dr. Manel Estellé. He's an MD and a PhD. He's currently the director of the Josep Carreras Leukemia Research Institute in Barcelona. Previously, he was the director of the Cancer Epigenetics and Biology program, also in Barcelona. And before that, he was a group leader at the CNIO. His early work has been critical for establishing promoted hypermethylation of tumor suppressor genes as a common hallmark of all human tumors. And his current research is devoted to the establishment of the epigenome and also the epitranscriptome maps of normal and transformed cells and the development of new epigenetic drugs for cancer therapy. He has an outstanding publication record with more than 600 original publications in peer-reviewed scientific journals, and also more than 80,000 citations. He's also a member of many international scientific societies, editorial boards, and reviewer for many journals and funding agencies. He has received multiple awards and recognitions. I really don't have time to go through all of them. And he's also a recipient of many important grants, as for example, and he has received an ERC advanced grant. He has been elected a member, last year he was elected a member of the European Academy of Sciences. And today's Manel lecture is going to be focused on the biology and clinical relevance of epigenetics in cancer. And I hope if he has time, he can. I hope he can give us also some clues about this emerging field of epitranscriptomics and how this is relevant and is offering new opportunities for cancer diagnosis, prognosis, and also for treatment. So Manel, thank you very much for being here with us today. We are looking forward to your talk.
Thank you very much, Maria Jose. I would like to thank the Instituto de Salud Carlos III and our friends and the International Agency for Research on Cancer for putting this together. And I'm very grateful for the kind invitation. So I'm going to give you like kind of an overview of the things that we have been doing in this field of cancer epigenetics, and as Maria Jose mentioned, a little bit about RNA modifications. That is the next thing together with epigenetics. And this work is done at the Josep Carreras Leukemia Research Institute here in close to Barcelona. And I hope I invite all of you to visit our place. I would like to start with this concept that I like very much. There's this concept of identity. And identity is identity of humans, identity of organs, tissues, and cells. This identity is in part, of course, determined by the genetic sequence. But this is just a part of the story. Because as you know, all our tissues and organs in a particular individual, they have the same DNA sequence, but they have different phenotypes. So it's not the genotype that in a person defines the phenotype. It's the regulation of the DNA, the RNA, and the proteins. And this is what we call overall epigenetics. And there are people that they like to meet to discuss what's epigenetics, but in a very simple manner, we can think that epigenetics are these chemical marks, and the two that are mostly there is an agreement that they are epigenetics are histone modifications, and these are chemical modifications of DNA. DNA measures 2 meters per cell, has to fit in a small nucleus, that has to be compacted, and compactation depends on the tissue. And these histone modifications that are methylation, acetylation, phosphorylation, etc., different amino acids, they provide these very complex histone codes that determine gene expression. But there is another chemical mark that has been mentioned before in the talk from my colleagues. This is the one that I'm going to talk mostly today. And it's the easiest one, it's the most genetic of the epigenetic marks. And it means that the C, the cytosine in our 4 components of the DNA, A, T, C, and Gs, this C can be from time to time methylated. And this methylation, the addition of this chemical group, usually is associated with gene silencing of the underlying sequence. It is critical for differentiation. And in fact, if we have an stem cell and we develop any tissue through differentiation, this is associated with particular chemical changes in DNA methylation and histone modifications. The same when we have a keratinocyte and we want to create a reprogrammable stem cell, an iPS. In this case, we go back in this DNA methylation and histone modification changes. All these remodeling factors, what they are doing really is changing the epigenetic scenario. There is a third phenomena that involves differentiation that is more new, you want to call it, that is called transdifferentiation. This is a cell type that originates another cell type without going through this intermediate stage of cell differentiation. Intermediate stage of a stem cell. Transdifferentiation is not something that we like to draw in a board, something that is theoretical, but it's something that is happening in real life in patients with cancer. And it's a way that many human tumors use to escape therapies. For example, a B acute lymphoblastic leukemia, under the pressure of the therapy against CD19, that's typical of these B cells, now can become an acute myelogenous leukemia, transdifferentiate to different cancer type. Another example can be a non-small cell lung cancer, under the pressure of tyrosine kinase inhibitors, now transdifferentiate to a small cell lung cancer that now is resistant to any therapy, for example, with EGFR inhibitors. This process of transdifferentiation involves many biological belts, but we have recently demonstrated that it is critical, this reorganization of DNA methylation patterns. And this is a model of B-cell acute lymphoblastic leukemia, degenerate macrophages associated with important changes in DNA methylation that happen in the promoters, but also at distant regulatory regions that we like to call, for example, enhancers. This is a high-C analysis to measure interaction between enhancer and promoters. In the BALL cell, there's no interaction, but when these cells became a macrophage, now there is this peak that shows the appearance of interaction between an enhancer and a promoter. And this peak appeared because the enhancer that here was hypermethylated now is hypomethylated, undergo loss of DNA methylation. This concept of identity in cells, as I mentioned, can be translated to humans. And we are different because we have genetic variants, single point mutations, copy number, etc. But we are different because we have also a polymorphic status of our DNA methylation. And this is inherited from our father and from our mother, the same way that is inherited classical genetics. And this was the first study that looking only at DNA methylation patterns, you were able to discriminate or distinguish three different human populations. Some of these variations in the DNA methylation setting can be associated with the risk of disease. Like this is like a SNP associated with the low penetrance risk of enhanced disease. And in this case, this is a methylation site that we found associated with the risk of developing colorectal cancer. One important difference between the genetics and the epigenetics fields is that when you have a mutation, more or less you die with that mutation, but epigenetics is more dynamic. Not only it's able to adapt to the environment, but it's able to be modified if we use drugs. Drugs that at the end of the day, what they do is they block the active sites of the enzymes that determine DNA methylation and histone modifications. So they are inhibitors of DNA methyltransferases, of histone methyltransferases, of histone deacetylases, etc., etc. These drugs not only are compounds that we can use in the lab or in a preclinical setting, but these drugs, after many years, have reached clinical stage. And now there are currently 9 drugs that we call epigenetic drugs that they have a clinical indication and they are given to patients with subsets of leukemias, lymphomas, and sarcomas. And these drugs are members of mostly 3 families: inhibitors of DNA methylation, inhibitors of histone deacetylases, and bromodomain inhibitors. And also there is also approval of histone methyltransferase inhibitors in a particular context of a subtype of lymphoma. But maybe the most known part of epigenetics in cancer, and has been mentioned also in the previous talk, is that many important tumor suppressor genes are silenced only by DNA methylation of this minimal promoter that contains a CpG island. And if you remember all these hallmarks of cancer by Doug Hanahan, that all cells, they have an inhibition of cell death, they have inhibition of an increase of angiogenesis, an entrance in cell cycle, loss of adherence, all those pathways can be driven by mutations, of course, on genes, but at the same time, they can be driven for particular methylation of genes in those pathways. And for example, this is an inhibitor of angiogenesis that is lost by DNA methylation. Then cancer cells, they have an increase in angiogenesis. This is icadrine. Icadrine, it's a gene that codes for a protein that glues together cells. And in cancer, breast cancer, there is DNA methylation and silencing of icadrine, and this is associated with the loss of adherence and the generation of metastasis. I want to give you the concept that is not only minimal promoters that can be altered by epigenetics, but also other regions. I mentioned before enhancers in the context of transdifferentiation, but also insulators that they counteract enhancers, and also cryptical promoters or internal promoters. They can be also the target of aberrant DNA methylation patterns in cancer. And the other concept I want to give you is that keep in mind that our genome is not really coding a lot of messenger RNAs, but it's coding more non-coding RNAs. It's transcribing more non-coding RNAs. These non-coding RNAs are many. You see here just ordered by size, the small one, the microRNAs to long non-coding RNAs. And my lab and others, these are just examples from my group, have been able to show in the last years that a lot of them are silenced by DNA methylation in human cancer. For example, the silencing of microRNAs, particular microRNAs, is associated with an overexpression of oncogenes or a degradation of tumor suppressors, etc. And the same happens with others like TUCRs, these long non-coding RNAs that are regulators at the same time of microRNAs. So this is a very complex scenario here. But it is true that at the end of the day, many times what we see in our faces and what we see in the cells, it's a change of the protein's expression. And an aberrant proteome is one of the characteristics of cancer cells, for sure. And this aberration of proteins can be at least in 3 flavors. You can have too much of a protein, too little, or you can have a different protein. And imagine that a gene amplification will give you this overexpression, gene deletion will give you this one, a loss of a protein, and a particular point mutation will give you a different protein. The same 3 scenarios can be caused by changes in DNA methylation. And just to explain you an example of this, we have identified that there is DNA methylation of the enzymes that are involved in the RAD pathway and the endoplasmic reticulum degradation pathway. So this is a way that targeting these proteins, you're able to induce more or less degradation of proteins. But another way to target protein expression is by targeting RNAs. And this is what has been mentioned before a little bit. If we think about DNA in humans, we have 5-methyl cytosine. And we have a little bit of 5-hydroxymethyl-cytosine, 5-HMC. This last mark only happens in some stem cells and sometimes in the brain, in certain periods in the brain. But that's it in DNA. But there's another molecule that is highly modified. And this molecule is RNA. Both non-coding RNAs and messenger RNAs, they undergo a heavy modification by different chemical marks. And this was thought to be like a one-way process, but in the last years has been shown that this is very dynamic. You get modifications and you can erase modifications. And here there are many. You can have methylated cytosine, like in DNA, but you can have also methylated adenine at the 6th position or at the 1 position. You can have pseudouridine instead of uridine, etc. So this is there are a lot of modifications here. And there is a constellation of proteins that take care of all this landscape. You have proteins that are writers, readers, and erasers of these chemical marks that regulate RNAs. And all these marks at the end are important in regulation RNA stability, RNA targeting, RNA translation at the ribosomes, etc. What's relevant, at least for my lab, is that in the last 5 years, we have identified a lot of genetic and epigenetic defects in all these enzymes and proteins that relates to this world of RNA modifications that, by analogy with epigenetics, we like to call also epitranscriptomics, whatever it happens over the transcriptome. For example, NUT16, it's an enzyme that is involved in RNA decapping, and this one is lost by DNA methylation. And this creates aberrant decapping, for example, in the MYC pathway. And it's important in TALL. This is an example, again, here of how epigenetics can change epitranscriptomics. DNA methylation is changing RNA decapping. This is an example of a gene amplification in another enzyme that modify tRNAs. This is a gene amplification in another one that's involved in RNA editing, A to I RNA editing, etc. And I want to give you now a few examples a little bit more in detail here. These are the enzymes and the proteins, and these are the chemical modifications that will be aberrant downstream in these tumors. This is one example of SELF2. SELF2, it's a gene that it's involved in splicing. And when we look at the splicing is like the biggest RNA modification. These are pieces of RNA put together, sometimes in a correct manner in normal cells and in a aberrant manner in cancer cells. And looking at all the proteins of the spliceosome, we identify that the one that is the most relevant to be silenced by DNA methylation is SELF2. And when you have SELF2 silencing, epigenetic silencing, you have changes downstream, like an increase in intron retention, exon skipping, etc. And this really creates a nightmare of aberrant transcripts for many tumor suppressors and oncogenes in breast tumors. Another example of alteration in epigenetics leading to epitranscriptomics alterations is this guy here, this enzyme, TW2. And this is an enzyme that what it does, it modifies a G at this position in tRNA, in transfer RNAs. We thought for many years that tRNAs were these boring molecules between there in the ribosome, just bringing amino acids to the messenger RNA, and that's it. But they are very more active molecules, and they contribute to the oncogenic pathways. And in this case, so normal cells are very happy because they have this enzyme that methylates this tRNA, and there is a very good binding between anticodon and codon in messenger RNA in the ribosome machinery, and the protein is happily translated. But it happened, for example, in colon cancer and other tumor types. is that there is DNA methylation and silencing of TWE2, and this silencing generates a defect in the modification of tRNAs. This tRNA cannot bind in the right way to the codon, and there is this RNA slippery sequencing, this ribosome frame shift, and at the end, there is messenger RNA decay. And there is a lot of loss of tumor suppressors here that they are caused by this previous DNA methylation and silencing of this RNA modifier. Another example is this one. This is another enzyme that modifies RNA. In this case, modify M1A in RNA. This is an M1A RNA demethylase. So in normal cells, this enzyme is expressed and remove M1A from certain RNAs. And then these RNAs are degraded, they are not translated eventually. But what happened is we have identified, for example, that in many lymphomas, like in Hodgkin, there is some silencing of this enzyme. And when this enzyme is silenced, the M1A chemical mark stays in messenger RNA. And when it stays, these messenger RNAs are very stable. And then you can produce, in this case, a lot of collagens. You don't expect to see collagens in a lymphoma, but this happens in 20% of this Hodgkin lymphoma. And these are this 20% of this malignancy that they have this poor overall survival. I want to move a little bit this very more research or preclinical concepts to using a little bit DNA methylation like a biomarker of response to treatments. It's what we call pharmacoepigenetics, trying to find the weakest spot in this cancer warrior. And this field started many years ago when we discovered that the silencing by DNA methylation of a DNA repair enzyme was associated with the response to alkylating agents in brain tumors. And this now it's a proof that is used everywhere around the world when you have a brain tumor to decide therapy. But there are other markers that can be proposed for that. Just to give you an example, as you know, in families with inherited breast and ovarian cancer that they carry BRCA1 mutations, it has been approved the use of these drugs that at the end damage DNA. They're called PARP inhibitors. But patients with mutations in BRCA1, germline mutations, are really a minority of breast cancer patients. But we identified years ago that BRCA1 is also methylated in around 10–15% of primary breast cases. And we have shown in vitro, at least, and in mouse, that these methylated cases that do not express the protein, they are very sensitive to PARP inhibitors. And they are sensitive at the same level that a mutation. So mutation and methylation, both of them are good predictors of response to PARP inhibitors. And this happened in around, as I mentioned, 10–15% of triple-negative breast cancer patients. I would like to mention and to introduce, of course, the idea that nothing is perfect here, and tumors are able to adapt. Tumors are small Darwinian walls that survive when we give them drugs. And for example, some tumors that are very sensitive to olaparib, PARP inhibitors, because they are methylated at BRCA1, they can become later resistant because that tumor in particular became demethylated. And this hypomethylation activates BRCA1. So, and the same happened for mutations. Germline mutation in BRCA1 can generate a different mutation later that creates resistance to the drug. So epigenetics is involved in generation of sensitivity, but also in the generation of resistance to drug, shown here by the shield of this warrior. One of the most famous slides for a while in the medical oncology field was this one, where you have this patient with multiple melanomas with a BRAF mutation. You give a BRAF inhibitor, great response, but the tumor reappear the tumor at the same places than here. This tumor is this tumor here, but it has a different composition, same geography, different demography, and it has acquired molecular changes to survive in the context of the drug. And this adaptation involves mutations, of course, but sometimes just involves DNA methylation changes. For example, these are tumors that are sensitive, colorectal tumors sensitive to oxaliplatin that damage DNA. And when we create a clone that is resistant to the drug, There are no mutations there to explain that, but there's DNA methylation and silencing of this gene, that it's a BRCA1 interacton, then it's a gene that participates in the response to DNA damage. So this is a marker of resistance to oxaliplatin and other drugs that damage DNA. Talking about this section about the real-life knowledge derived from epigenetics, one thing that we realize, of course, and has been shown also by expression previously, is that not all tumors are the same in a patient, and there are different types of cells there, for sure. And this was one of the first approach that we did a few years back, just to show that, for example, in colorectal cancer, we take different sections from the same tumor, and we analyze DNA methylation. So 90% of the sites are the same content. We have this 10% of sites that they have different DNA methylation values, showing that there is an heterogeneity within the tumor. And this heterogeneity can explain resistance to therapy. I would like to give you this example that goes back a little bit to the beginning of my talk, because it's an example of application in real life of the knowledge in epigenetics and this concept of identity, how DNA methylation is very good to provide identity. And there is in cancer a clinical situation that we call cancer of unknown primary. So it means that it's a patient that the first sign of the cancer, it's a metastasis. And the medical oncologist is unable to find the primary site, cannot find the origin of the tumor. This is a big problem because this patient has then extremely poor prognosis. It's a disseminated disorder, and you don't know what to do in these cases because you don't know the primary site. So what we did is to take a picture of the DNA methylation profiles of these tumors and compare this picture with our collection of more than 10,000 human tumors to see how it looks. If this looks like a pancreatic cancer, a breast cancer, a colorectal, a biliary cancer, etc. So we can give a name to this unknown metastasis. And this has changed, was changing also some of the outcome of these patients. Another application of a clinical application or translational application of this knowledge in epigenetics, it's the use together with the new immunotherapies. Immunotherapies has represented a revolution of the last years in cancer treatments, but it has several problems. For example, even for the tumors that has been approved, like in melanoma or non-small cell lung cancer, many of those patients do not respond. In lung cancer, more or less, around 1 in 3 patients respond to immunotherapy with immune checkpoint inhibitors, anti-PD-1 or anti-PD-L1. So what we did is using a typical model design study, using a discovery cohort and a validation cohort, we studied a huge collection of non-small cell lung cancers treated with anti-PD-1, trying to see if we obtain a profile, a signature of DNA methylation that predict response to anti-PD-1. And we obtain the signature, epi-immune signature. If you are epi-immune positive, it means you are a responder to the immunotherapy. You are epi-immune negative, then you are not going to respond at all to the therapy. These people here with epi-immune negative, for example, we know that they have an enrichment in fibrosis in the tumors. And in these guys here, they have already some degree of activation of interferon gamma pathway. You can combine this with other markers. For example, if a patient is epi-immune positive, PD-L1 positive, CD8 high infiltration, a high mutational load, this is a patient that is going to respond very well to the therapy. But if you have a patient that is epi-immune negative, PD-L1 negative, CD8 negative, very few mutations, this is a patient that immunotherapy will be a waste of time and money. So we have to think about options for those cases. And I would like to mention that this is not only immunotherapy for drugs, but epigenetics can help in the immunotherapy for cells. Because as you know, the newest therapy for cancer involves cellular therapies. And this is patient 0, started with this patient 0 by Emily Whitehead. this girl here, she was diagnosed with a B acute lymphoblastic leukemia, and here she had a few weeks to live, really, and she was saying goodbye to her puppy here. She received, T cells from this patient were isolated and were re-engineered in the lab to fight against positive cells for CD19. And these CD19 cells are usually the B lymphocytes. And they were given back to the patient. And now, this is 2020, but now 2022, she's perfectly fine 10 years later. So this patient has an official cure using this therapy, using these T cells. That overall, we call this therapy CAR T cell therapy. And this is great news. But the problem of all these therapies is there are several. One is this is still very expensive. So it would be nice to have a rationale which patients should receive the therapy. But not only they are very expensive, but sometimes they have adverse effects, the development of cytokine release syndrome and effects at the level of the neurological side effects. Not only that, but some patients do not respond to therapy from the beginning, and we don't know why. So we wonder if we can try to find markers of response to this cell therapy using epigenetics. And this is what we did. We put together an academic trial from Barcelona, here in Catalonia, and Spain, in Israel, by the Sheba Medical Center, and in Italy. And we studied a huge collection of B-cell malignancies that has been treated with CAR T cells. And we look at the epigenetic profile of the CAR T cells just before given to the patient, just a few minutes before it's given to the patient, to see what is the composition of DNA methylation there. And from this, we obtain two signatures, the signature EpiCAR positive and EpiCAR negative. EpiCAR positive, it means that those CAR T cells are going to be very effective. These are EpiCAR T cells that were transfunded to the patients. They're going to have a huge clinical effect, while EpiCAR negative are going to have a very limited effect in the patient. We can wonder what's in these signatures, and we have done this by flow cytometry and analysis, and we have seen that these EpiCAR positive cells, they are enriched in less differentiated cells. Well, EpiCAR negative, the ones that don't work very well, they are more committed T cells. So it looks that these EpiCAR cells, more differentiated, they have a short lifespan in the patients. And probably this can explain why they have reduced efficacy. While these more immature cells, they will stay longer, and there will be this long-lasting effect to kill the B cells in these patients. This type of experiments and data, it's supporting this concept that now there are many clinical trials trying to combine immunotherapy with these drugs that we call epigenetic drugs that you mentioned before. And there are many reasons for this: to increase viral mimicry, to increase the expression of antigens, to select the right population of T cells, etc. And you will see that a lot of these clinical assays now are being developed, combining anti-PD-1, anti-PD-L1, an inhibitor of HDACs, or an inhibitor of DNA methylation, etc., etc. So summarizing my lecture, I just want to say that the lab has a strong interest in providing not only knowledge of what's wrong in the DNA methylation in cancer, by providing biomarkers that can be used in the stratification of these patients, providing clues about overall prognosis, but also clues about sensitivity to drugs. And these are just a few examples from the last years. And I would like to conclude mentioning this example, because, of course, as you may realize, like a lot, epigenetics and cancer and DNA methylation to study, but I understand this is just a part of the big problem of cancer. It has to be combined with other omics, and together this multi-omics can provide very important insights for the biology of cancer and for the future treatment of cancer. And this is a study that we did together with the National Cancer Institute, that I like it very much, that we put together DNA methylation, expression, mutation, copy number, immunophenotypes, etc., in a particular subtype of human tumors, that are tumors from exceptional responders. And these are people, they have overall a tumor that is very advanced. So according to statistics, they should live only a few weeks more. But when the medical oncologists give the classical drug, no new drug, the classical expected drug, they have these amazing responses, and the tumor almost disappears. So what's happening here? So we can think that this is a miracle, or we can think that there is a molecular explanation for this. And what we did here is put together this huge collection of exceptional responders and did all these multi-omics that I mentioned before. And the conclusion that we had is that at the end, these tumors that respond very well, they are in patients that are lucky. Lucky because despite having an advanced disorder, they have changes, defects in the same pathway. And when you are given a drug for that pathway, that tumor disappears. Let me give you an example. Like this is a GBM, glioblastoma, stage 4 glioma, one of the worst tumors you can have. You expect a overall survival of 1 year and a half maximum. And this is one case, for example, that this patient was more than alive after 7 years. So how is possible? And when we analyzed this patient had 2 mutations in DNA repair enzymes and a methylation in ATHR1 DNA repair. So when this patient receives a drug that damages DNA, that tumor cannot survive. That tumor cannot find routes to escape from those drugs. And that's the reason that tumor died. And that patient has overall survival. And for me, this is a good example of the power of how we can translate epigenetics, epigenomics, together with other omics for the benefit of the patient. And I would like to finish here. I would like to recognize, of course, the relevance of the genome, very important, but what underlies beneath, at least, is equally important. And this is the epigenome, and now also the epitranscriptome. You will see many new findings about the epitranscriptome. Just to mention that just 2 weeks ago, the first patient treated with an epitranscriptome drug entered trial. So this is going to be a new field, a new game, and hopefully we'll see a lot of interesting findings. I would like to finish here. I would like to thank all the members of my group for their hard work and all the sponsors of my lab. Thank you very much.
Thank you very much, Manel. That was very interesting. There are no questions yet from the audience. If you want to ask questions through the chat, or you can raise your hand.
So, in fact, I have a question from Senko, from Senko and all the people that presented from that part. So I like it very much. This question about for example, the predisposition in leukemia, in childhood leukemia, no? The epigenetic predisposition. So what are the thoughts about that?
Well, thanks, Manuel. That was really impressive presentation. And regarding the question, well, the idea is that at the birth, in this neonatal blood samples, we can see something, as I said, that may be a signature of past exposure during utero life. We show some amazing examples, I think, and you see this AHRR, aryl hydrocarbon receptor repressor, that has methylation. P-value is 10 to the minus 200. That with that confidence you can say that this is a smoker. So this is very clear that we can, looking at the epigenome, we can identify some kind of footprint or exposure. But then now, whether this or can constitute a risk. So this may not constitute the risk of future cancer development. So we started with the idea that we can actually identify signatures of exposures, but also markers of risk, predictor of risk. And so this may not be necessarily linked to the known exposures. Smoking clearly is not, we did not see that this HRR, for example, is associated with risk. But we say maybe there is something in the epigenome, because as you said, the regulating transcriptome and the proteome and so on, it's actually epigenome is critical for gene expression program. So, and this is stable. I mean, otherwise it's not epigenetics, as you said. I mean, this is stably propagating the gene activity states. So the idea is that to see something early on that may constitute risk. And actually what I showed briefly, in my slides, we have now a manuscript submitted showing consistently in different cohorts specific genes that whose methylation actually are associated with the risk of leukemia. So it's a risk. So it's not like some diagnostic marker, that can, you say this is really, but just kind of associated, robustly associated with the future risk of leukemia. Now we also there are some interesting genes maybe we can discuss. It's not published yet, but yes.
And this marker, it happens during pregnancy, or is inherited from the father and the mother?
So no, this was identified in cord blood, in the cord blood of the baby.
Yeah.
And so we did not look actually whether this is we don't have actually material of the parents. And often these are the well powered study, prospective study. So regardless of, but we believe that maybe some of them might be linked to the so-called imprinted genes. Some of those genes. But not all. And then actually what we also looked at the diagnosis. So we look at diagnosis, whether you can see the same thing, what we see at the birth. So it's well ahead, years before the diagnosis. I mean, usually peak of leukemia, childhood leukemia is about 3 years and 3 years old. And so we looked at whether we see something or that actually stays in anti-diagnosis, and whether this event itself may predict the outcome. Yeah. So that's the actually quite interesting question, and I think we have I think quite solid. We try to publish, but we have a quite solid I think finding as a proof of principle.
Yeah, thank you very much. And yeah, Maria, if you allow me, I have one for Giri.
Sure.
Giri, so I like it very much the toxin in the Burkitt lymphoma. So in Burkitt lymphoma, then what is the connection? So have you or somebody have proved how the toxin really changed downstream DNA methylation? This is something that happened mediated by any protein, or this is a direct action on the DNA? How it works?
It's a question for Rita, right?
No, it was when you. Sorry, maybe it was president. Rita, so
Rita presented.
But no problem. Yeah. But it's fine. Actually, no problem. We do have some studies that were done on cohorts linking the exposure to aflatoxin actually to some changes in methylation. So we do have some signature changes related to mycotoxin, and we are trying to see in vitro as well what is really happening. About the mechanism, we did not look at this, but some studies actually link studies how the metabolites of those toxins might impact some epigenetic regulator that later on will deregulate the methylation.
Understood. Thank you.
Yeah. Right.
May I just, okay, so there I see some hands up, but if I can just maybe ask Manuel quickly. I mean, this impressive, study on lymphoblastoid, I think published 2013. I was always puzzled by this. I think this was a really smart approach, because difficult to collect blood from across the globe, but there is in your bank or whatever. Biobank, you have collection of lymphoblastoid cell lines that are immortalized by EBV, right? So, and this is a nice source to study, because these are nearly normal cells, right? And then also representing different origins, ethnical, and so on. So I was quite struck that although this was 450K arrays used, I mean, this is quite good, robust, well covered, genome is well covered, but the differences are in the methylome are really small.
Yeah.
I mean, I don't know how to compare, for example, SNPs versus, that all these African Americans versus Caucasians and so on. The differences are really small in methylome, in the peripheral. These are peripheral blood derived, cell lines. Yeah. Although you did find, colorectal cancer linked to, some, but on a global level, I think that it was remarkably similar between the, distant, let's say, populations.
So we have, as you look at DNA methylation we do in humans, for example, there are some that are a lot. The differentiation is big, but these are in a small case, in a very few sites. A lot of this big difference, they have downstream a type of genetic variant. For example, a defect, a SNP in an enhancer, this creates a big methylation difference in the enhancer. And this separate populations also. But this it's a minority of the whole. But in this case, the difference in the methylation is big. But in these cases, they have usually an SNP or a mutation downstream in the underlying DNA sequence. When you look at the remaining cases, you see the difference, but as you mentioned, they are small. And in fact, this is one of the richness of epigenetics, because in mutation you have the SNP or you don't have it. It's 0.5, 1. So that's another story. Here we can be more versatile about this. And this is enough. And imagine that somebody expressed the gene instead of 100%, express 80%. That particular, and that 20% difference, it's enough. Or even this 10% difference, it's enough to have a different, for example, penetrance of disease. And this can be difference of expression that, as we said, 10, 20% maximum. Because if you express more or less, probably you have an adverse, a really bad disease, and you don't survive. So you need the right program in there. So you only allow some degree of difference, but not too much, because then you'll not be human probably, or you'll have a disease.
There is a question from the audience. I think there is a problem with the chat. So I think some people couldn't write in the chat, but I got a question through the email, and I think it's for everybody. So they're asking, how can we be sure which is the normal epigenome? Because at the end, the exposome that alters the epigenome is continually changing, and today the exposome is very different from what it was 200 years ago. Even short periods of time through our life can change radically. So even epigenetic changes will be different in different cells of the organism with the same exposome. So what is your question? Yeah.
So if you allow me, before Senko and the others can go in. So I will ask first, what's the normal genome? I'm not sure it's the normal genome. Because now that we do deep sequencing and more people are sequencing the world, all of us are mutants. All of us has an important germline deleterious mutation. All of us, one or two or three or more, probably. Okay. So even the genome that should be more stable is highly variable, and you don't know what's the normal genome at all. But we reach a consensus. This is a normal genome. Okay. We do an average, and this is a normal genome. For DNA methylation, for example, that the field I know the most, so this is the consensus. Say we analyze 1,000 people, healthy, okay, for this cell at this level of resolution, and this is the normal epigenome. But when we go to single cell, now we can go single cell epigenome, and then you have a different normal epigenome. Because before you had many cells together, but now you can do different cells. And the other question is, and this is a difference with genetics, that this is more, it changes more than others. So you have to define always the normal epigenome for that age and for that condition. And this is the normal epigenome. The normal epigenome you expect from a newborn baby is not the same normal epigenome that you expect from a person that is 80 years old.
Yeah, I think this is well explained, but I just want to add that the problem or confusion comes that the epigenome is tissue specific or cell lineage specific, and often until recently we are analyzing often population of tissue or population of cells that are heterogeneous, and therefore but there are reference epigenomes. If you take the I don't know, if you purify T cells, whatever, you will see that the epigenome would be pretty homogeneous. And often the problem comes because of the changes in subpopulations, even the blood and so on, and because each subpopulation has a specific epigenome, then there is heterogeneous kind of we are averaging, but as Manuel said, now if you look maybe a single cell level, although even then you will see that different, maybe individual cell might have a slightly different, but it will be pretty much more homogeneous in terms of epigenome. So yeah.
More questions?
I have maybe nothing from the audience. If I may, so just coming from the angle of chemical carcinogenesis, so I was fascinated by the concept of the epitranscriptome, and this is all very interesting as an innate or intrinsic process, dynamic and so on. But then I was wondering also if what would be Manuel's thoughts about additional changes introduced in the RNA by, for example, chemical adduction, which would also impair the function of the RNA. And one example would be temozolomide treatment that you mentioned also for the glioblastomas, right? Temozolomide has a very strong genotoxic, alkylating effect. Is this also happening at the level of RNA? And I don't think this is studied sufficiently. There's few studies on RNA adducts, but nothing really, broad or systematic. Any thoughts on that?
Yeah, so this is a great question. It's something that the field now, epitranscriptomics in cancer is a very young field. So, and the things that we have done in genetics and epigenetics now are going to be translated to epitranscriptomics. And when you mention it's a necessity in the field to know about these things. But I think it's going to happen. I think that a lot of carcinogens are acting on DNA, some on proteins, but some are acting on RNA. And there are some data even before showing that some mutation in RNA finally give a mutation in DNA, for example. So it is possible that, imagine that a change in RNA finally induce a change in DNA methylation or histone modification in epigenetics. That makes sense.
A lot of crosstalk, basically.
Crosstalk all the time. And because some of these marks, they share the same donor, S-adenosylmethionine, we're talking about methylation, okay? Or acetyl-CoA, if we're talking about acetylation, not only of histones, but now RNAs are acetylated. So it's possible. The same chemical marks we saw in DNA and histones, we're seeing a lot of them in the RNA. So a lot of this can relate to carcinogen exposure, food, nutrition, so many of those.
Exactly. And I think it can be maybe extended even beyond the RNA world, again, to other macromolecules in the cell. If you think about electrophilic compounds, they can be attacking lipids, structural proteins. And
yes, absolutely.
It's the totality of the damage, really.
Yeah, but in my case, I think I'm going to stick for RNA for a while. It's too complicated. Yeah,
it's already way too complicated.
It's too complicated. Yeah.
Okay, so hopefully we will get some interesting insights soon. Thanks very much. Yeah.
So Rita, you have another question?
It was about epitranscriptomics as well, because it's a fascinating field, I find it. And I was wondering about the knowledge on the heterogeneity of the epitranscriptomics as well, and how the technical challenges is limiting so far our knowledge about this.
Yeah. So there is this feeling, this sensation that epitranscriptomics is more dynamic and more heterogeneous than, for example, than epigenetics. Was more dynamic and more heterogeneous than genetics. And epitranscriptomics even more, let's say, okay, and faster to change, okay. So you require some time to change DNA methylation in a cell. You require less time to change RNA modification profile. But the data are starting to emerge now about this. We know very little about, for example, memory of neurons. This relates to M6A RNA. Probably it does, but nobody has done. Aging, it relates to M6A, this kind of things. People have started now finding the enzymes, looking how this is a dynamic, a reversible process, because before it was thought it was fixed, and now it's dynamic. Okay, go and go back. And now what they're studying has been in the last years, the mutations, defects in cancer of this, and what happened downstream. But this now is going to be changed to all the environment, and that you have an interest there. Environment is going to change a lot of these patterns for sure. And heterogeneity, yes. Single cell epitranscriptomics, even single cell epigenetics is starting now. So single cell epitranscriptomics, I think there are 3 or 4 papers. And that's it.
So we hope for the best. Yes, and regarding the treatments, I had one small question about your opinion, Manuel, about the targeted CRISPR editing versus those treatments that are not specific.
Yeah. So let's say the target CRISPR also here is like all the gene therapies. If they have to work first for the mutations. If the mutations they work first, then we'll go there and we'll do for the epigenetics and all that. but so far even in the genetics, they're counting problems. But I would say that they should start with the monogenic disorders for sure, and how they are doing, blindness, metabolic disorders in kids, and from then we can move to the others. I really like the idea that sometimes the drug can be inspecific, okay, but the patient is specific, and the patient has something that for that drug is the right patient, a translocation, a profile of DNA methylation, something that even the drug is so inspecific, but only those cells has the better.
So the effort should be made on profiling the patient to decide a better treatment.
Absolutely, unless you have a more specific drug that's very difficult to get.
Yeah.
There are patients that respond the best. Even a pancreatic cancer, there are 1% that respond a lot to a HDAC inhibitor. But why is it responding so well? Because maybe it has a mutation in HDAC7, I don't know.
Yeah.
And that can explain that particular patient.
Thanks.
I have one question for Zdenko. You mentioned that the reversibility of the obesity changes, the epigenomic changes. So that got stuck in my mind. So can you elaborate a little more? And also I wanted to ask you if you are doing something with the reversibility, for example, of aging epigenetic changes, or if you're doing something about it.
Yeah. Well, epigenetic, that's the defining feature of epigenetics. So in contrast to genetics, epigenetics are irreversible in principle, although we know that this is not, this is not practically that simple. Finding
when you do an intervention, when you do the weight loss.
So the idea is that we have examples that or studies showing, for example, that the epigenome pattern are kind of specific with some conditions. For example, obesity. In obese individuals or metabolic disbalanced individuals, you have a specific pattern of the epigenome in the even surrogate tissue, in blood. This may be cause or consequence, we don't know. But anyway, it is specific for some conditions, health conditions. Now, the idea is to see if these persons undergo some kind of treatment or interventions that they would lose weight, they would do exercise, or they would have some kind of treatment for metformin that is known to kind of be used, for example, in even survivors or people that have a high risk of, and so on. So the idea is to have a different interventions and see whether the epigenetic changes that are associated with these conditions would be reversed. And that's what we took advantage of. The big two studies, actually, we did one in Europe, one in the US. With metformin, but also this is really a well-controlled, placebo-controlled trial. And so we did the methylome, but also we did other omics layers. And we see something. It's too preliminary to say, but we think that this type of studies, intervention studies, are really unique to address this, to see to what extent the modulating lifestyle, for example, modulating doing exercise or losing weight and so on, can reverse these changes. I think that's real. That's one important question, because of course, apart from therapy, far from what Manuel talked a lot about, epigenetic drugs, here is more like modulating by adapting the healthy lifestyle, which of course has importance for cancer prevention, but also for this in the survivors. As you know, we often talk about prevention or treatment, but there is a huge population of cancer survivors, or whatever they call it. Some of them, they don't like to be called survivors anyway, but people that actually are successfully treated. Although we know that many of them don't have really normal life anymore, but then there are consequences. But nevertheless, they are successfully treated. And many of them are at risk of secondary cancer or recurrence, right? Many of them, and that's a big problem. And often the therapy is not efficient, because there is a Darwinian evolution of tumor cells, and therefore there is really interest to have some kind of adapt the lifestyle that would minimize the risk of recurrence. And therefore, what we did in this study was on the survivors of breast cancer that are metabolically, affected. And that's what we are now trying to analyze and see what's going on. But at the moment, we don't have yet, story. But this study is ongoing.
Yeah. So, Zdenko, how close are we to really being able to tell somebody, this is the way that you should change your lifestyle according to your genetics, epigenetics? Many years to come? Is it close? What do you think? It's a difficult question, but
I think we talk a lot about personalized, therapy, but also personalized, prevention, and that's something that is we should aim at. I think for some cancers, for example, for breast cancers, probably we advanced the more than elsewhere, because that's the common cancer. There's a lot of studies. There are, of course, screening. There are different models to deal with the people at high risk. For example, germinal mutation and so on. So there are different models to kind of estimate the risk and how to minimize the risk. So I think for some cancers there are things advanced quite well, but in general I think we are not there yet, and we feel that the epigenetic markers could be part of this risk assessment. We know that the screening, for example, of course, screening is important. National screening programs are important, but I was shocked even the top reference persons would argue that some of these screening do not change anything. I don't know whether this is wrong to say, but they say that it was Marmot what was talking about, that the breast cancer screening doesn't change the outcome. That if you look without screening programs, there are no difference in terms of either mortality or whatever. I was shocked. I don't know whether this is whether this evolved, but what I'm saying, screening of course is important, but apparently it is not without a lot of, it's a costly, it's a disadvantage, there is, and there is a damage, there could be potentially harm and so on. So there is need, to incorporate other, other advances in science and biomarkers. For example, if you look at the prospective, I did not have a time to talk about, for example, if you look in prospective blood samples, we can see something that can predict risk. I'm not talking about diagnostic markers, predicting something risk using artificial intelligence and all these algorithms can predict the risk of breast cancer 15 years before, or risk. So we are not there yet to incorporate this in some kind of model that you can sell. But I feel that we are advancing, but maybe we are not there yet to really to say that we can have a more kind of really personalized advice. Yeah. I don't know, Mane, what you think, but
so I think that going back to the screening, I think screening is extremely important, has been very useful, but it has to be a good screening. So, for example, cervical cancer screening.
Yeah, of course.
90% of the cervical cancer, people in Spain used to die by cervical cancer, all the female. And now almost nobody dies by cervical cancer in Spain. No? But it depends on the type of technology and what can we do later. And this is the question here with breast cancer, for example. I think in colon cancer, the screening colon cancer is working well also. But we have to see the impact later. And related to Pilar's question about the obesity things. So as you know, Senko, people with cancer and obesity, they do worse than people that without obesity in cancer. And it is said that there is a crosstalk between the fat cells and the cancer cells, a chemical crosstalk between them, and probably this can involve epigenetics, and we have done some work with collaborators about this, looking for the profiles in the fat that is close to the tumors to see if there is something wrong there, the same in other microenvironment cells.
Okay, somebody was asking, they are sending me the questions. I think there was a problem with the chat, sorry. Is about the proteomics. They insist and they will incorporate the epigenetics study with the proteomics, but I think you mentioned that, no, Manuel?
Yeah. So as I said, proteomics, it is great. It's difficult to do in a clinical setting, to do proteomics. You have to do protein markers, no? So you do a multiplex immunohistochemistry, at the end is a proteomics, very short proteomics, but it's a way to screen. But I believe that in this multi-omic analysis, proteomics should be there. And in fact, we're doing now the proteogenomics studies of BLL together with the NCI, and we're using sequencing, mutation, and proteome and phosphoproteome. Should be there. Keeping in mind that our genome the proteins is a part of what they produce. Okay, most is non-coding, but it's too complicated to analyze. But at the end, it has an impact on the proteins. Yeah, many times.
Okay, I think Manel, you have another meeting right after this one, no?
Yeah, unfortunately.
So unless there is something really important that you want to remark, any of you, please go ahead.
So I would like to say that these are great meetings, and they should be enhanced in the future, this type of collaborations between us and our French colleagues, and I will be happy to collaborate in any way that you want.
Always pleasure to exchange with you, Manel, and I agree that this kind of bilateral meetings are really falling nicely into our mission, and that's promoting international collaboration. So we are in France, but we have an international mandate, a global mandate, and having Spain not far away. So obviously this can reinforce, and I think that's I would really encourage Pilar and Maria and others to continue.
Yeah, Zenko, I also always say that there were very few Spanish people in your teams, the national teams, and very few Spanish grantees there at IARC. So that's what my mission, to try to change that. And that's the mission of this conversation. So they are going to be there in the web, so everybody can go. I will send you the link so that you can refer it to other people to go to the seminar and watch it again, or watch it for the first time. So thank you all for your time, for your precious time. I really appreciate it. Thank you, Maria Jose, for being here with me. There will be another meeting in January that will be about the environment and how it affects also cancer, with Joachim Schutz. Yeah. I hope to see you again. I hope to see your new center also very soon.
Yeah.
And thank you all for being there. And goodbye.
Thank you, friends.
Thank you, friends.
Thank you.
Bye.
Bye-bye.
All the best.